This year's Alamo Advocate Program is dedicated to the woman who founded ABCF and the Advocate Program, Dale Eastman. We are delighted that SABCS has also dedicated the entire week to her. It is now time to start our program. Michelle Tregear, Chief Programs Officer for the National Breast Cancer Coalition, will be introducing the speakers for the Project Lead Advanced Topics Program, Revolutionizing Breast Cancer Immunotherapy, Harnessing Macrophage-Targeted Therapies.
Welcome, everyone. The National Breast Cancer Coalition is pleased, once again, to have you here and to open up with the Alamo Advocate Program. We've been doing this, I think, and I can't even remember the first time. It's been quite some time. And we do have a very special, exciting scientific program for you. But I don't want to waste too much of our time. So what I'd like to do is just introduce our advocate moderators, because they are going to introduce our speaker. So Nancy Poorvu is a longtime breast cancer advocate whose work is shaped by her experience as a survivor and a person living with metastatic disease. A 2016 Project Lead graduate with updated training in 2023, she founded the Soulmates Peer Support Program at the Dana-Farber and has been an active member of the Metastatic Breast Cancer Alliance and the Dana-Farber Cancer Institute advocacy community. Nancy serves on multiple research communities, including the Department of Defense Breast Cancer Research Program Review panels, and for the Dana-Farber Cancer Institute SPORE program, bringing a strong commitment to patient-centered, scientifically rigorous research. Our other advocate moderator today is Susan Koegel, who is a biomedical scientist and education turned breast cancer research advocate shaped by her experience in a high-risk breast cancer family. She is a 2024 graduate of NBCC's Project Lead and now serves as the co-lead for the Dana-Farber Cancer Institute Breast Cancer Research Advocacy Group. And I should say both of them have worked with our speaker today as advocates, which is one of the reasons why I thought they would be perfect match to partner with Dr. Guerriero. So Susan contributes broadly across the Dana-Farber and to the translational cancer research, well, the TBCRC. Working groups and reviews, and then she reviews applications for the Dana-Farber Cancer Institute as well. She is dedicated to strengthening communication between researchers and the patient community to accelerate meaningful progress. And so right now what I'd like to do is have Nancy come up here. She's going to start us off with a moment of silence, which is how we always launch every single activity that we run.
Lauren Bezanson was a dedicated, longtime NBCC field leader and advocate. She was diagnosed with breast cancer in 1989. At the time, she had limited treatment options, and following high-dose chemotherapy with an autologous bone marrow transplant, she knew there had to be a better way to treat breast cancer. She decided to become an activist to help make a difference. In 1999, Laurel joined as a founding member of the Maine Breast Cancer Coalition and served as the advocacy chair for the organization. Through her work with that group, she attended the first NBCC conference in 1993. She became an early member of the NBCC and served as field coordinator for Maine and later graduated from Project Lead. In 1993, she represented her organization at the White House when NBCC delivered 2.6 million signatures to President Clinton and asked for a national plan to end breast cancer. She attended many NBCC summits and lobby days over the years and was always advocating for NBCC's legislative and public policy priorities in her home state and in Washington, D.C. Some of her most recent advocacy included placing a letter to the editor in the Portland Press Herald, which helped secure the support of Senator Collins on the Metastatic Breast Cancer Access to Care Act. Laurel passed away on March 30th, 2025, of metastatic disease. Please join me in a moment of silence to remember Laurel.
Great, thank you again for joining us here today for this special session. We're thrilled to be joined by Dr. Jennifer Guerriero to explore potential new areas in breast cancer immunotherapy. To set the stage, Dr. Guerriero will first give a brief overview of key elements of the immune system that are relevant for today's talk, and then do a deeper dive into exploring the potential of tumor-associated macrophages. Nancy and I will then sit down with Dr. Guerriero to explore how she has worked with advocates and what we have brought to the table in her work in order to highlight potential roles that we can play as advocates in the research process. After that, we'll open it up to questions. So now a all too brief introduction of Dr. Guerriero. Dr. Guerriero earned her BS in biochemistry from Northeastern University on a pole vaulting scholarship. Had to add that. She also earned her PhD from Stony Brook University in molecular and cellular biology and immunology and pathology before doing her postdoctoral training at Dana-Farber. She is now an assistant professor at Harvard Medical School and a lead investigator at Brigham and Women's Hospital, and her lab really explores the accelerating the potential of immunotherapy in the treatment of cancer. As a faculty member with the breast oncology program at Dana-Farber, she's also focused on understanding sensitivity and resistance to immunotherapy. So Dr. Guerriero, thank you for joining us.
Okay, well, thank you so much for the opportunity to be here. Okay, looks great. And we'll use this microphone or? Okay. So I am going to give a brief overview of immunology related to immunotherapy of breast cancer, and then go into some new work that my lab is working on to overcome immunotherapy resistance and chemotherapy resistance. So before I start, I want to take the opportunity to thank everyone here for their advocacy and awareness for breast cancer. This is my grandmother, who was diagnosed with breast cancer the year I was born and lived with metastatic breast cancer my entire life. This is a picture of her and me at my PhD celebration, my graduation. She was a true advocate of breast cancer. She did everything she could to be connected and to advocate for other breast cancer patients and their families. And truly everything that I do and aspire to be is because of her. So I thank all of you as well for everything that you are doing for this field. So immunotherapy, I think, has been an incredible blockbuster across most solid tumors. In 2011, we had our very first approval of immunotherapy. Since then, there's been 149, so 150 FDA approvals of immunotherapy across all cancers. In 2023, it was considered the breakthrough of the year. In 2014 and 2015, there was an acceleration of approvals by a number of teams across the country and throughout the world, really, resulting in the Nobel Prize in 2018. Now, a lot of the success that we saw early on was in a disease that was previously thought to be incurable, which is metastatic melanoma. So before we had approvals for immunotherapy in metastatic melanoma, these patients were treated with chemotherapies as the standard of care. And the median survival was about 6 to 9 months. The 1-year survival, about 30%, and 5-year survival was really sort of unheard of for metastatic melanoma. Now, with a number of new immunotherapies that have been approved for metastatic melanoma, we're actually able to cure a handful of these patients with 10-year survivals approaching over 40%. So really significant therapeutic responses in the setting of melanoma, other diseases as well, such as lung cancer. So the premise of immunotherapy is that we activate the immune cell called a T cell. And I know many of you already know what a T cell is, but I'll go into a little bit more information. So in this movie here, the T cells are in red, and they're kind of floating around. The cancer cells are in green. And if you, can you see my arrow? Maybe not. Oh, there we go. So these are the cancer cells. You can see this one's green, but this T cell here is attached to it. It's just killed that cancer cell. So the premise of immunotherapy is that a T cell is capable of killing a cancer cell under certain circumstances, which I'll detail in the next couple of slides. And so getting these T cells to kill cancer cells is the goal of immunotherapy. But cancer cells are very smart. They can make things expressed on their surface that prevents the T cell from killing them. And we call these checkpoint blockade signals. So this is like a cartoon just for simplicity. So this is a T cell. This is a tumor cell. And this tumor cell has upregulated this protein called PD-L1. It's smart. It doesn't want to be attacked by the T cell. And so that signaling, that killing ability of the T cell is blocked. It can't do it. It's turned off. So immunotherapy blocks that inhibitory signal. So we like to say, and I'm sure you've heard this, that immunotherapy takes the brake off the T cell so that the T cell can then kill the cancer cell. To make this a little bit more complicated, the T cell here is in an off position because its PD-1 and PD-L1 are interacting. So this is the tumor cell, and this is the off switch here. So targeting PD-1 or PD-L1 then turns the T cell on. There's one other really important signal that I want to point out in this slide because it is a theme throughout this talk. So the TCR, this is the T cell receptor. The T cell has to have a go signal, which we just talked about. It also needs to be able to recognize the T cell, the cancer cell. Otherwise, the T cell would be in your body killing all kinds of cells. That's not what we want. We want it to be a direct attack of the cancer cell. And so this T cell receptor binds to what's called this MHC class 1 domain on the cancer cell. That's a go signal. In this particular example, we have a stop. But then you add the immunotherapy, and you have a go signal. Go for the recognition of the cancer cell through the T cell receptor, the TCR, and MHC class 1, and then you've inhibited either PD-1 or PD-L1 with immunotherapy, and that T cell kills the cancer cell. It's much more complicated than even that, and so this is not for you to really read through today and find detail. I think we'll be able to provide the slides for you to look at afterwards, but there are a lot of stop signals. There are also a lot of go signals, and so there has to be a delicate balance in mediating this. One, to prevent autoimmunity, right? You don't want your T cells killing everything in your body, but in a certain context, you want your T cells to kill cancer cells. And there's a number of new novel agents that are being explored for immunotherapy approaches, ones that both activate the go signals, ones that block the stop signals, and then other ones that further enhance the entire picture of this immune response, which we'll get into in a second. So in breast cancer, as of just 18 months ago or so, there was over 330 immunotherapy trials that are in breast cancer that have targeted accrual of more than about 50,000 patients. So that's is shown here in this figure, looking, a number of them are in phase 1, phase 2, phase 3 trials, very few actually in phase 3, but those are the dark blue. They span over adjuvant, metastatic, neoadjuvant settings. And this is really, again, just for you when you're at home to read in more detail. But the different strategies of immunotherapy, are they the checkpoint blockade that we just talked about, PD-1, PD-L1? Are they cellular therapies, which we'll actually discuss in the second part of my talk? Are they monotherapies, combination therapies? And so a number of trials are being explored in the context of breast cancer in many different settings. However, today we have 2 approvals for the treatment of breast cancer. So we have, sorry, this is not very well projected. In the early setting, we have neoadjuvant pembrolizumab, which it blocks that PD-1 signal on the T cell to activate it to kill a cancer cell. And then in the advanced setting, we also have pembrolizumab, which is used in combination with chemotherapy. So of the 150 approvals across of all different types of cancer, only 2 are for breast cancer. So why is that? Well, if you want to target a T cell with your therapy, then you must have T cells in your tumor. And breast cancer is notorious for having fewer T cells than other types of cancer. So this graph here at the bottom on this x-axis shows all the different types of cancers. And then this y-axis is an enumeration of the frequency of these CD8 positive T cells. So you'll note that these cancers over here on the right, these liver and kidney and melanoma, they have more T cells in the tumors than, for example, breast cancer, which has, as I mentioned, fewer T cells. So this may be a reason why there are less responses in breast cancer compared to other types of cancer for immunotherapy. Now within breast cancer, the different subtypes, there's also differences in immune cell infiltration. So this graph shows the number of lymphocytes. These are your T cells and B cells. These are your cancer-killing cells. So HR-positive disease has the fewest T cells out of all of the subtypes of breast cancer evaluated here. This is triple-negative breast cancer here on the right. And of course, as we know, the data I just showed you, the 2 approvals are in triple-negative breast cancer. So why do we not have T cells in breast cancer? And this is what my lab thinks about a lot. How can we get T cells there? How can we keep the T cells in the breast tumor? How can we get them activated in a way that makes sense for breast cancer, which might be different than other types of cancer? I'll note that we have spent quite an enormous effort trying to understand the molecular regulation of T cell recruitment in HR-positive disease. And a very talented fellow in my group, Dr. Kenichi Shimada, will present today in the Best of AACR session at 2:30. And he'll give a more detailed description of the molecular understandings of why HR-positive disease may not have T cells. Just as a summary of that work, and maybe a teaser if you want to go see the talk, we and others have shown that HR-positive breast cancer has a downregulation of this MHC class 1 protein. That's the protein that binds to the T cell receptor to be recognized. So if the tumor cell can't be recognized by the T cell, we think the T cell just leaves because there's no reason for it to stick around. So this is actually independent of these PD-1, PD-L1 interactions. So I'm detailing here in the summary scheme that HR-negative disease, let's call it triple-negative breast cancer, has this MHC class 1 molecule that talks to the T cell receptor on the T cells, but when that's absent, the T cells are no longer present. And we can see this morphologically when we stain breast tumors with either looking for the nucleus, so all cells are shown in blue, or this MHC class 1 molecule. So in triple-negative breast cancer, lots of the cancer cells have this MHC molecule. That makes sense. There's more T cells there. However, over here, the estrogen receptor positive tumors actually have no expression. That red on there is actually staining immune cells, not cancer cells. So we've been able to validate this using morphological features as well. And to summarize sort of this HLA and T cell infiltration and responses to immunotherapy, what we see is that the frequency of this is the subtyping of the different breast tumors. You can see the aggressiveness, and again, this is sort of just a cartoon to summarize larger picture ideas here. The effectiveness of immunotherapy is dictated here, and then this correlates with the presence of immune cells. And actually, T cells are a predictive marker of responses across tumor types. So I've talked a lot about T cells. I know all of you know there's more than just T cells in the body, in the immune system. And so to step back a little bit and just do a quick immunology or immune cell 101. So all immune cells start from a stem cell, hematopoietic, multipotent stem cell that can become all of your immune cells. And so this hematopoietic stem cell can become these lymphoid progenitors, which give rise to natural killer cells, your T cells, and your B cells. These are called your adaptive immune cells. We'll talk about that in a second. On the other side here are your common myeloid progenitors. These become your basophils, neutrophils, eosinophils, macrophages. Those are your innate immune cells. To understand the difference between innate and adaptive immunity, I've used this schema here. So adaptive immunity, T cells and B cells, they're a slower response to insult, but they have memory. So once they identify a cancer cell in your breast, they then have memory of that tumor cell, and they can hopefully, ideally, find it in a metastatic niche. These are different than your innate immune cells. Your innate immune cells are your first line of defense against bacteria and pathogens and allergens. And so they respond quickly, within minutes sometimes, within hours. There's no memory response. They're just ready to go, ready to kill bacteria. Similarly, they can also kill cancer cells. And so it's the two systems, the innate and adaptive immune responses, that work together to ensue a functional, durable anti-tumor response. So I wanted to go through the cancer immunity cycle just briefly. There's a lot here, and I'm sure many of you know this cycle. This is a very famous figure that has had many different versions of it redone. This is the newest version. It's from 2023. It's the Melman and Chen review on the cancer immunity cycle. If you haven't read these papers, I mean, you should read the newest one because it has the most current data in it. But actually, it doesn't change much. What we know about immunology of cancer hasn't changed in the last decade. So I just want to walk you through the key steps that are required for the complex ability of a durable anti-tumor response. So first of all, you have to have antigens, pieces of the cancer cell that the T cell can recognize. So what happens is the innate immune system takes up pieces of the cancer cell, shown here. It travels to different parts of your body to find T cells. So for example, in the lymph nodes, it presents these antigen to the T cells and educates them and tells the T cell what it needs to kill, right? This is very important to make sure that the T cell is not killing all of your cells in your body. So the innate immune system educates the T cells, which is shown in here. The T cells then traffic through the body. They get to the tumor, which is shown here. And the T cell now looks for things it recognizes, such as cancer cells. Once it recognizes the cancer cell, it can then kill the cancer cell, and this cycle continues. Now, we talked about go and stop signals. There are many through this path. I mean, it's a miracle this even works, right? All the go signals, all the stop signals. And so all of these complexities have been investigated and evaluated, and this understanding has led to new biology and new therapeutic tools. I won't go into all of these. But I just want to summarize some of the factors that may prevent T cells from killing cancer cells. So the first is a lack of these innate immune cells, dendritic cells or macrophages, that educate T cells to teach them how to kill cancer cells. Lack of tumor-specific proteins, or we call them neoantigens, that are available to educate T cells. I just showed you in this last slide all of these GO stimulatory signals. They're needed, they're required. And in some cases, there's a lack of these signals. The loss of MHC class 1, I highlighted that earlier in my talk when there's the T cell can't recognize the cancer cell. High infiltration of cells that inhibit T cells, like macrophages, which we'll talk about in a second. And then a number of unknown factors that are continuing to be discovered. So I want to leave some optimism in targeting HR-positive disease. So I mentioned that a senior fellow in my lab will be giving a talk later today about the mechanisms by which HR-positive tumor cells may hide from T cells. But actually, we know that HR-positive disease has some responses to immunotherapy. There was, in the I-SPY trial, this was a trial that evaluated pembrolizumab, so that anti-PD-1 molecule, in stage 2 and 3 HR-positive disease, and found that adding pembrolizumab to chemotherapy demonstrated small but significant benefit. More recently, in the Keynote 756 trial, demonstrated, so the Keynote 756 trial demonstrated that, I'm so sorry, I've lost my pointer, it's there. Okay, demonstrated that the addition of pembrolizumab induced a significant benefit for more early stage patients. And so I think there's great optimism here in utilizing immunotherapy in HR-positive disease. And so when we think about what can we do in this setting? How can we enhance T cell infiltration, T cell activation across breast tumors, but especially HR-positive tumors that we know have low HLA signaling, low T cell numbers? What are the mechanisms that we can employ? And so this schema right here summarizes a number of the investigations that are ongoing to enhance T cell trafficking. And some of them include cytokine delivery to activate T cells within the tumor. Some of them include inhibiting those suppressive factors, inhibiting macrophages, for example, which I'm going to get into more. In terms of other cells, so we've talked about T cells for the last 17 minutes or so here. This schema right here shows a estimation of different immune cell types across all cancers. So here on the x-axis, these are all different types of cancers. And then all these colors represent different types of immune cells. So the macrophages, for example, are shown in this tan color. Macrophages represent up to 50% of the tumor and can be quite suppressive. They can inhibit T cell function. And so over here on this analysis, there is this team who did this analysis, then correlated all these different types of immune cells with how patients did clinically. And what they found, again, when they looked at both breast cancer, which is shown here, and lung cancer, which is shown here, the cell types, which are all abbreviated. We immunologists love to abbreviate. We like to use multiple names for the same cell type. It makes it much more complicated. So ignore the abbreviations and just take my word that these are all macrophages. So macrophages are the cells that correlate most significantly with a worse clinical outcome. But it's not to say that all of these cells in the tumor are probably doing something and likely working together to coordinate the immune response. So what I'd like to do now is to jump into some of this new macrophage biology that my lab is starting to uncover, where we think that T cell biology is very well understood at this point. There's other immune cells in the tumor microenvironment that we think can be targeted, that can be harnessed for anti-cancer therapies. So what I'm going to do is I'm going to walk you through some data that we've, or some work that we've done together with Dana Farber and the breast spore and our support from the patient advocates. And then I want to show you some new data, first-in-class therapeutic strategy that we've developed that we think is going to be quite powerful. So of all the things I've done, I'm most proud of these collaborations working together with basic, translational, clinical teams. And so as an overview, I of course want to acknowledge these people up front for their contributions. So first I'm going to tell you about how we've identified a specific type of macrophage. It's called a lipid macrophage. I'll tell you more about it in a second. So we identified these cells. We then identified that they play a role in therapy resistance, and we've identified ways to target them to overcome resistance. And then I'll present to you this first-in-class, this brand new strategy. I've actually only presented it publicly twice, so this is my third time, so this is very exciting. Okay, so I showed you this schema. You're convinced macrophages make up a huge portion of solid tumors, but I want to show you that visually because I always think the visual representation is the most convincing. This is a normal breast tissue, just a regular non-cancerous breast tumor. This is all human data. On the bottom here, this is a breast tumor. The dark brown staining indicates macrophages. So you can see 50% of this tumor is full of macrophages. Looking at these survival curves, we know that patients who have more macrophages, so that's these in the green, have a worse relapse-free survival and a worse overall survival. So why is that? Well, I always like to, I told you a few slides ago that these innate immune cells, these are T cells, B cells, monocytes, macrophages, we all have them everywhere, in every tissue, in your blood. They're not specific to cancer. One of the jobs of macrophages, in addition to fighting bacteria and pathogens, is to facilitate wound repair. So if you have a wound, this is my schematic of a wound, monocytes are recruited to the tumor. So monocytes are the cell that travels in your blood and then becomes a macrophage when it enters the tissue. So the monocyte senses there's a wound that needs to be healed because the dying cells secrete all of these signals that I need help, I need to be healed. So the monocytes come to the tumor, they become macrophages, they secrete factors to help the tissue repair. Well, in cancer, the exact same thing happens. A tumor is just a tissue, and it is a tissue that has low oxygen and poor nutrients and sends out all these signals to your body so that these monocytes come in. And to no fault of the monocyte, because it's doing exactly what it's supposed to do, it repairs the tissue, because that's what a monocyte does. A monocyte macrophage repairs damaged tissue. So in the setting of a tumor, when it does this, it secretes factors that help the cancer cells survive. These cells secrete factors to help with angiogenesis and metastasis. These cells recruit suppressive T regulatory cells. And importantly, they inhibit the cytotoxic T cells that we want in the tumor to kill cancer cells. So because of this, there's a number of strategies that have been developed, that have been tested preclinically and clinically to inhibit all the things I just talked about. It's the molecules the macrophages are making. It's inhibiting angiogenesis, inhibiting T regulatory cells. But this is like sort of chaos, right? And maybe inhibiting one thing is not the right strategy. So my lab is always focused on the central cell, the macrophage, targeting the macrophage directly and eliminating it or changing it. And so we've devised a number of strategies, which are shown here on the left, that have moved forward in clinical trials and are being tested in clinical trials and additional preclinical strategies. But we really wanted to know, this cell here, what is special about it? Can we directly target that cell? And so we took a step back a few years ago, and we decided to make an atlas or an encyclopedia of the immune cells in tumors. I'm going to just highlight the macrophage part of it because that's the focus of this work here. So we took two strategies. One is we took data that's already been published, and we mined it, and we annotated it, and we put it all together. And then the other thing we did is we used an in-house cohort of patient samples that we performed single-cell RNA sequencing on. So all this data is of single cells, so we can look at the DNA of every single macrophage and understand how they're the same and how they're different. This is a UMAP. I'm sure you've seen UMAPs, but basically what a UMAP does is it takes all the data, right, all of the data, and puts it into a nice little schematic where the things that are close together are similar and the things that are far apart are different. So for example, these orange macrophages and these blue macrophages are probably more similar than these down here, but they're a little bit different because they did separate on this, this high dimensional projection. And so what we did, and this is a little bit complicated, I'm aware, is that we can then look at the genes they express. We can look to see, did this macrophage correlate with good T cells or bad T cells? Did it correlate with no T cells? And each of these is a piece of the puzzle, because what we really want to know is which are the bad macrophages and how do we target them? So by doing all of this, we're able to generate this encyclopedia, or this atlas. And what I want to draw your attention to, because I skipped a lot of details here, but I promise, actually, this is on bioarchives. I can give you the citation if you want to go back and see the paper. These two populations, GPNMB, you don't really need to know what that is. It's the highest expressed gene of this macrophage, and these SPP1 TAMs. These were the two most suppressive macrophages. These are the worst macrophages in there. They're inhibiting T cells, and biologically, they're called lipid macrophages. They're taking up fats, they're making the whole tumor become suppressive, incapable of allowing T cells to infiltrate. And when we look at these two subsets of macrophages that we were very excited to find, we find that indeed we can look at their gene signatures and we can try to understand how are they signaling, what are they doing, and what we identified is exactly what I just told you. They're involved in lipid signaling, T cell inhibition, and hypoxic regulation. So it's helping with angiogenesis, all the bad things. So we found our bad macrophages. Really interestingly, and not surprisingly, we're not the only ones to identify lipid-associated macrophages as bad players in the tumor microenvironment. Prostate, lung cancer, colon cancer, across the board, these macrophages are found. Work that we'd done at Dana-Farber, we'd actually already identified that lipid macrophages were a major problem in PARP inhibitor responses. So several years ago, we had identified that PARP inhibitors, after PARP inhibitor, lipid macrophages infiltrate into the tumor, and they are associated with poor response to PARP inhibitor therapy and T cell exclusion. So this is mouse data. We have an amazing mouse model of BRCA-deficient triple-negative breast cancer. When we treat these mice with the PARP inhibitor olaparib, you can see here in brown there's macrophages, but after therapy there's huge recruitment of new macrophages. And what they express are molecules suggestive of lipid metabolism. CD36 is the lipid receptor, so these take up lipids. CSF1R and TREM2. These are key characteristics of bad macrophages. So this was very exciting because we can target these therapeutically. But does this happen in patients? So we are, again, very fortunate to have such a collaborative interdisciplinary team across the Harvard system. So we worked with Harvard Medical School, Dana-Farber, my lab at Brigham, to look at patient samples, so samples from patients pre and post PARP inhibitor, to see did the patients have increase in lipid macrophages. And this data is a little complicated, but the answer is yes. After PARP inhibitor responses, patients have macrophages that indeed have the same signature. So we were very excited because this is a targetable cell. So CSF1R, right here I showed you, this goes up on macrophages after the PARP inhibitor. So there's drugs against CSF1R. So we thought, let's go forward with CSF1R. And not to belabor this, but anti-CSF1R therapy is approved for 2 different indications, not for breast cancer, but it's approved for the treatment of tenosynovial giant cell tumor, which if you see here, this is the tumor. It is full, all the brown staining, those are CSF1R positive macrophages. After treatment, the macrophages are eliminated, shown here in the bottom pictures, and patients derive great benefit. More recently, CSF1R therapy was approved for graft-versus-host. So we went back to our mouse model to test this hypothesis, to see if anti-CSF1R could eliminate these lipid macrophages, to enhance PARP inhibitor responses. It does. So shown here in the graph, there's the untreated mice are in the red bar. They succumb quite quickly to their disease. You see there's a prolonged survival with PARP inhibitor. When we add the anti-CSF1R agent, you dramatically extend this by 100 days. So that is quite significant in a mouse model. By getting rid of these bad macrophages, we got more T cells, activated T cells, which is exactly what we want. So we've moved forward to a phase 1b trial, again led by my colleagues at Dana-Farber. We worked very closely with our patient advocacy team for the biopsy and the dosing of this combination. So it's the first time axatilimab, which is a anti-CSF1R agent, has been combined with olaparib. We're already half enrolled, which is really exciting. We have biopsies from baseline. We have biopsies from after PARP plus the CSF1R agent. All of them are in my laboratory. We will do all of the biology and mechanistic studies to understand what happens when you get rid of these suppressive macrophages in this PARP inhibitor setting. And we have gone on to obtain more funding to be able to do this analysis. And so, this collaborative nature of working together with our advocacy team and the clinicians and my translational team has just been wildly successful in being able to move these sorts of things forward. And I want to tell you about a new strategy too. So this is very exciting. The trial data looks very exciting. We're adding actually another 10 patients to this, and potentially adding another arm for some other, based on some other data we have. So going very well. What we really wanted to understand is how else can we target these cells? And so when we went back to these two types of macrophages that we had identified from our single cell analysis, that encyclopedia that we made, what we noticed is TREM2 was uniquely expressed by these bad macrophages. And I already mentioned others have already found TREM2 in other cancers. So these are all different cancers: skin, liver, lung, breast, bladder, colon. Brown are TREM2 positive macrophages. So they're in all solid tumors. They're not in normal tissue. So normal tissue doesn't have these bad tumor macrophages. We also know that if you have TREM2 in your breast tumors, your outcome is worse. Preclinically, others have evaluated blocking TREM2. I know this is a lot of data. If you use anti-TREM2, you get a reduction in tumor burden, which is here in this purple, compared to no treatment. And if you add PD-1, you even get a complete response. Similarly, in lung cancer, this is a mouse with lung cancer. All those purple nodules are lung tumors. If you treat with an anti-TREM2, if you block TREM2, you reduce lung tumor burden. There's 2 trials testing antibodies against TREM2. So this is a plausible target. But we wanted to try something a little bit different. And so we proposed something called macrophage replacement to eliminate these harmful TREM2 tumor macrophages. So the goal is to get rid of these TREM2 positive macrophages. They express TREM2 right here. So what we've done is we've engineered human monocytes. We take blood from the patient, take out the monocytes, and engineer them with what's called a chimeric antigen receptor. So that receptor can identify these TREM2 macrophages and eliminate them. So it might sound like science fiction, but actually chimeric antigen receptors are just T cell receptors that we talked about before. So T cells, and this is why I tried to emphasize this before, T cells express these T cell receptors that bind MHC class 1, right? That's how the T cell finds the cancer cell. In the binding, then the cancer cell dies. This has been wildly successful for engineering T cells with these chimeric antigen receptors to kill cancer cells. However, it's only been successful in the context of blood cancers, not solid tumors. So CAR T cells have not yet been successful in solid tumors, but again, have revolutionized the field of blood cancers. So why? Why can't T cells get into tumors? Well, we already talked about that. That was my whole first part of my presentation, right? T cells have a very hard time getting to solid tumors. Why is it? Well, they're not recruited in high numbers. They're probably inhibited by macrophages. And so what gets to the tumor? Monocytes. So we have changed the strategy. So as opposed to using the T cell as the effector, we've changed it to monocytes. So monocytes, they go right to the tumor in high numbers. So we've engineered the monocyte with this receptor to target bad macrophages. And so when it does that, it then becomes a macrophage itself, but a good macrophage. And we've engineered it to secrete factors that then induce further T cell activation and tumor cell killing. So as proof of principle, and I wanted to show a little bit of high-level data just for anyone who might be super excited about this. So as proof of principle, I know you're all excited about this. So as proof of principle, and I just have 2 more data slides I want to show you. We made mouse, CAR, monocytes, and we made human. And what we needed to demonstrate in a cell culture dish was that these CAR monocytes could kill the target cell, which are the bad macrophages. And so indeed, if you take this monocyte and you express this CAR, oh, I'm sorry, you can't see my pointer. So if you take this monocyte and you express this chimeric antigen receptor, it can then recognize TREM2 and gets rid of these macrophages. So this is a measurement of the ability to kill the target cell in the murine system and also in the human system. So we can kill, in a cell culture dish, these bad macrophages with our CAR monocyte strategy. I know this is wild, right? Because you're taking a monocyte and killing a macrophage. And even when I give this presentation to scientists, they're like, wait, you take macrophage, kill a macrophage. So this is a very complex concept. But we put this in mice to see if this would induce tumor reduction or enhance survival. And so again, you have your CAR monocyte. We take the monocyte out of the mouse, engineer it with this receptor that then recognizes these bad TREM2 macrophages. After 14 days of treatment in this murine, this mouse breast cancer model, you have, this is the tumor volume of just untreated mice, just mice with tumors. If you just inject monocytes into the mice, the tumors are about the same, but you can reduce the tumor burden by half using our platform. Really importantly, we can induce 100% survival with this strategy. So we have gone out much further than this 20 days. I have to admit that this is what's in the patents. This is sort of, what's publicly allowed to be shared, but we've combined with PD-1. We've gone out further. We're only using 1 million cells. This is a very, very low dose of cellular therapies. Billions of cells are being used for the CAR T cells. Why? Because CAR T cells don't get to the tumor. CAR monocytes do. So this is very exciting. So we have 100% survival of mice here. So just to summarize all of these findings, and so we are moving forward with this. We're spinning out a company. We're moving fast toward IND enabling, where we think that this is going to be a true new therapy that will have maybe not monotherapy for approval, but getting rid of those bad macrophages, we think, is what needs to be overcome to get the T cells activated. All right, so just to summarize here. So I hope that what I demonstrated was that the intentional analysis of pre and post therapy samples was really what got us here. Understanding what's in the tumor microenvironment, and we had to step way, way back, right? We had to do single cell analysis of many, many biopsies to really understand what are the bad macrophages and how are they interacting with T cells, and how can we find a therapeutic target for to benefit patients. We think that the lipid macrophages are indeed not only the predominant immune cell in primary and metastatic tumors, but these are the bad guys. Why do CAR monocytes have so much potential? I try to emphasize that monocytes rapidly go to tumors in high numbers. They also survive. T cells don't survive in the tumor, but monocytes do. And so getting this therapy that's in a monocyte to the tumor, we think is the right strategy. And we think that this is a platform that is going to enhance endogenous T cell responses. So I'll stop there. I'm happy to take, I think we're going to do a panel first and then questions after that. Okay.
Well, thank you so much. I mean, that was a fantastic talk, and I just felt like I learned so much every time I hear you explain your work. Today, we want to think with you about why you think it's important to include advocates in your laboratory work, and what are your thoughts about how and why including advocates enhances your work?
Yeah, well, we treat mice, so a little bit different than patients. And I do admit that even though I'm so connected to my clinical collaborators, I do still sort of have that narrow, oh, this is a mouse, right? Let's do this with the mouse. Let's treat the mouse with 12 different drugs and do 6 different biopsies, and that will translate just fine to the clinic. That's not true, right? So even in our preclinical designs, we need to think about what is the strategy that will be most clinically relevant. So that's the first thing is, you know, it's a blessing that we are able to have interaction with patient advocates because they shed so much light onto what is meaningful science. And then the other place where it has been truly transformative is designing that clinical trial, that AXLAP clinical trial, because the patients are receiving olaparib as standard of care, and then we're asking them to add a drug to that, this AXLAP drug. And it's, as you know, it's been approved. I showed 2 different indications, so it should be safe. But then we wanted them to come in for that infusion, and then we wanted them to come back for a biopsy, and then another blood draw. And I remember you guys saying, can we do this on the same day? I mean, did the patients have to come back 6 times this week? And so if we expect that patients are going to want to be part of a trial, we need to think like a patient, right? And so we've had huge success in enrolling this trial, I think because it's conducive for the patient, right?
Right. I agree. One of the most exciting times as an advocate is at the very beginning of our work together. And I think we'd love for you to comment on what makes that beginning of the relationship work well for you, and how it works with an advocate at the beginning when you're just meeting.
Yeah. I don't need to tell you guys this because you're on the other side, but researchers often approach advocates the day before grants due. Right? You know this. I don't need to tell you this. You already know this, right? I need a letter tomorrow for my DoD grant. And who are you? What do you work on? So that's not our approach. And I think after you do that, you know, the investigator learns, wow, actually this is a very meaningful relationship and I should have started months and months ago. So I love the same thing. This CAR monocyte project, as a brand new project, yes, we were submitting grants well ahead of the deadline. We were reaching out to advocates for their input on the aims and the concept and the meaningfulness of the science related to patient care. And that first meeting is so fun, right? Because we go through why do we think that this is the next best thing, right? Mm-hmm. And help us form that concretely to present it in a grant or a paper or a presentation. And having an advocate be as excited about the science as you is really just like truly a special thing.
Right.
Yeah.
So I think the best advice that I have for advocates who are meeting a researcher for the first time, no matter how new or how experienced you are, is to really try to come in at the same level as your researcher and open yourself, talk freely, ask as many questions as you can. Don't be intimidated by the research or the researcher, but really aim to understand the research and to realize that you have your lived experience and that is very important, what you're contributing. That what you're contributing is very important.
Yep.
And means so much. And that there's so much more you can do than just writing that letter of support. And the researcher may not know what it is that you have to offer. So you want to really educate the researcher about what it is that you do. That give the menu of what services an advocate provides. And I try to really lay it out. Here's what I can do over the course of your research project. I will do work with you on your plain language summary.
Yep.
I will write your statement as part of your proposal. I will read the patient advocate portion of your proposal. I will then help you with dissemination of your results. I will work with you on a poster. I will,
Yeah.
and I give the whole array of what services I can provide.
Yeah, I think
for
those
you.
are really important. The thing I really like that you said at the beginning was, step up. Don't worry if you're asking a question that you think might be naive. It's probably not. And if you have that question, probably somebody else will too. I'm always so impressed with the questions that advocates ask, and sometimes make me realize I have a hole in my description here. And as a reviewer of many, many grants, I often rely on that lay abstract, especially if I'm not an expert
Right.
in the exact thing this is on. I go to that lay abstract, and a lay abstract is best written with a patient advocate.
Absolutely.
Yep.
Absolutely. If a researcher says, I will write your letter of support for you, decline that offer. That's not a good offer.
Yeah.
You want to know enough about the research to write the letter of support yourself.
Yeah, absolutely. I think that, builds on the point about, you're running a whole laboratory, and you talked today about all the people that are training in there. And we've had the pleasure of working with some of those people. You've taken to not only, incorporating advocates in your work, but really having that as an element of training the next generation. And I think this is a big opportunity because the earlier in their career that people are working with advocates, the more likely they are to continue that as they move forward. So how do you incorporate that when you're working with some researchers that might not have envisioned that they'd be engaging with advocates?
Yeah, I think you're right. I think any trainee in my lab works with an advocate, and we try to, think about what's the expertise of the advocate and sort of their background and what disease setting makes sense. And was this advocate an engineer, and might they know a little bit about engineering of immune cells or something that would be exciting to them to contribute to. But I would say everyone comes into my lab not without that sort of on their radar.
Yeah.
And so it's a simple concept, right? And it makes a lot of sense. And I think that as projects unfold and my trainees start to work with advocates, it becomes very clear that this is a necessity for the research. And it just becomes part of what we do, right? And I think that institutes like ours are very good at that. I think that it's them then going out into the world and continuing with that mindset and continuing to engage these foundations and Komen, who do a really nice job with that too, to continue. Most of my trainees are interested in lab-based positions like I have, and so they will, I think, most benefit from these relationships, and it's, sort of every generation will expand, right, in their progeny of those with that similar mindset. So, that's how I try to teach that within my team.
Yeah, and I think it's great as an advocate to work with a young investigator, an early career investigator, because it's so natural to have that conversation of, have you ever worked with an advocate before? And the answer is usually going to be at that point, no. And so you really get to have that conversation about what does advocacy mean to you, us as an advocate, you know, all the questions. And I can also, you know, see the relief on the person you're asking that question, because they don't know what to do either, right? If they're beginning in that relationship as well. So to have that conversation and put it on the table, and I really can expand the opportunities moving forward of having people reach out far beyond, you know, the proposal at hand.
Yeah, and you know, Liz has been part of my Komen career development plan and all that PARP inhibitor studies for over a decade. And so, you know, it's really sort of formed the path of that from really preclinical discoveries all the way through to the clinical trial, which is part of our SPORE now.
Right. And you know, I acknowledge that at Dana-Farber, we are very fortunate to have a culture of using patient advocates, and that isn't the culture everywhere. But I think if it isn't the culture where you work, it's important to get educated through Project Lead as an advocate, and we are going to have further training about the advocacy relationships and how to form them, and to be able to go to the leadership and make us, you know, to really make a case for how patient advocates can be useful, and the whole, as I said, array of things that we can do that are helpful beyond letters of support, and the way that we've really educated ourselves so that it is, you know, almost a professional, you know, position.
Yeah, I think, you know, thinking about, you know, what you just brought up about the Komen, this having an opportunity as your career extends to work with someone in the long term is great for an advocate to have that relationship for a long time, but also I'd imagine it's helpful as your research changes and expands. So what has that experience been like working with someone for over a decade?
I know. I definitely remember the first time we talked about the PARP inhibitor project, and again, I was very naive. I think I was a postdoc then when I first. I was a postdoc. Yeah, I was in Tony Latai's lab, working with Jeff Shapiro too, and really thinking a lot again about mouse models and how the mice respond to cancer as my primary focus. So that was eye-opening, right, and important, and that became a major focus of my lab. It's now our SPORE project, so it's really expanded, right? The Jeff Shapiro, who is at Dana-Farber, and Philippa Lynch, I had their pictures up there. She's running that phase 1B trial. The three of us just got an R01 to study the biopsies in it, and so, you know, it starts very small. I was a postdoc, like, working on macrophages in response to PARP inhibitor, and a huge team comes together to push these things forward, and yeah, Liz has been there since day one. But I think also, you know, it was important. I presented always at their patient advocate meetings or journal clubs, and I know, you know, I hope you guys learned a lot, but I learned a lot too, right? The questions that are asked, you have to ask questions. Don't hold back, really. Because those questions help me reframe what's important, right? Help me not be a mouse doctor. Help me make a drug that goes into the clinic, right?
Right.
Absolutely. Yeah. Right. Should we open it up?
Yeah, I think we should.
All right. We thought this would be a good time to open up to questions online or in person. If you can go to the microphone so we can hear you, that'd be great. I think it might need to flip on.
Hello.
There
okay,
you go.
You can hear me now. Hi, I'm Marilyn Miner. I am a Project Lead mentor, and so I'm also Project Lead graduate. The concept of patients being involved in the research and working with scientists came from the National Breast Cancer Coalition. We all know that, right? And many of you in the room are all Project Lead graduates. So it's more of a statement than a question. If you want to be involved, we clearly know that there are research scientists out there who understand the value we bring. I will tell you, not all of them do, as you said. I had someone ask me last year to be on his project and write a letter, and I thought, no, I can't do that.
No.
So, you know, he was a bit surprised. He did submit. He did not get his proposal approved. No kidding, right? He didn't even know what a project lead or even what an advocate really does in our first meeting. So I say all of that to say, understand the value you bring and look at the massive number of people in this room and in your state and across the country, quite frankly, who you can reach out to get information about it. We're all going to be intimidated until we do it.
Yeah, right.
That's just a given. So the majority of people with whom I've spoken about it, that's the first thing, and even for me, that was the first thing I said. I'm intimidated to work with a scientist because I don't know the science, but that's not why I'm there.
No.
Right? That's not why I'm there. So I want to encourage all of you to be willing to sign on, and most importantly, use your project lead community to help you get where you need to be. Okay?
Thank you, Marilyn.
I like that a lot.
Thank you for that support. Perfect.
It should be on.
I don't know. It's on? Can you hear me? Okay.
Hi.
So I want to go back to some science a little bit because so my claim to fame is I was cured using tumor infiltrating lymphocytes by Steve Rosenberg.
Amazing.
Yeah. But I'm like So I'm sorry, Judy Perkins. I'm with the Alamo Advocate Committee here. I work with the National Breast Cancer Coalition. I've been through Project LEAD. I've been around thanks to Steve Rosenberg for the last 10 years. But so my question is, I'm still a unicorn. Like I really thought a bunch more people would be following me through this door, and it hasn't worked. And I'm hearing what you're saying. I know there's a researcher at the NIH. She's working on these genetically engineered myeloid cells.
Rosie Kaplan.
Yes, Rosie Kaplan. And
She calls them gemmies.
Yeah, gemmies.
They're engineered monocytes as well.
Exactly,
Yep.
the gemmies. And I'm just like, obviously we need a freaking cocktail because the T cells
Right.
by themselves. I mean, I had the T cells, I had some pembrolizumab, which handled the IL-2 breaking system. And like, how do we get what you're doing and what Rosie's doing, and it all needs to get into the IV bag? Like, you guys talking to each other?
Yeah, Rosie's amazing. But great point. So so Rosie's amazing. Great colleague, great friend. I have a number of her trainees who work with my trainees. It's a little bit of a different strategy, but sort of a similar concept. You're right that it is going to have to be some sort of combination where T cells and macrophages are probably modulated. I'm working with a number of T cell CAR companies where we may put them both together in that bag and put them into the patient, right? And it's not, obviously mindful of just putting as much as we can into the bag and infusing it. I think it's right, though. What you're saying is that it needs to be very mindful, right? We know we need to get T cells. We know we need to get rid of macrophages. And so can we combine those strategies to do that? I think that we can. And then, of course, kill the cancer cells. So I don't have the answer for you today. We have a number of hypotheses that we're pursuing with different drug companies, starting our own company, those sorts of things. But
I'm just curious, like how it would even fit in with the thing I went through. They give you high dose chemo to completely knock everything out.
So you don't have to do that with the CAR monocytes. For the TIL therapy, you do. So TIL therapy is basically they've found the good T cells in your tumor, expanded them, and put them back in. And remarkable success in those that it works in.
Melanoma.
Some, yeah.
In those that it works in, it's remarkable, and then it's not everyone, right?
Right.
So
But like your thing, so if you did that thing, it would kill all the macrophages and everything, but then they would come back online like a couple weeks later.
Just kill the bad tumor macrophages. The regular macrophages in your body don't express TREM2. Does that make sense?
But you would deliver it later when your immune system came back. Like if you killed everything with the chemo, then gave the TIL, and they also give you IL-2,
Yep.
then like,
I'm
just thinking hypothetically, like later you would maybe give the pembrolizumab and the other stuff to I don't know. Just
No, I love all of this thinking. You should come to my lab meeting. Actually, you should have patient advocates at your lab meeting.
I think so too.
I love this. It helps with the language in the lab meeting. It has, amazing ideas. You're right. I don't know the answer, and all of those things need to be considered. The advantage of the CAR monocytes is that there is no lymphodepletion. You take the monocytes from the patient, you infuse it. I envision it in combination probably with a PD-1 agent or with a CAR T against the tumor. So I don't see it with chemo right now.
We're a little above people.
Oh, sorry. Okay.
But
Hi, I'm Cheryl Jernigan. I'm from Kansas. That's a nod back to Jane. I'm a longtime advocate. Was diagnosed with what we now know as triple-negative breast cancer 30 years ago, but we didn't have HER2 then. So anyhow, I've learned from Liz Frank, your best, and I've learned from the gal behind me, Jane. But I want to say to your this isn't why I came up here, but to your comment about having advocates in your lab, go for it. We do that at KU, Kansas University. Cancer Center, and our advocates love it, and the basic scientists love it. They have people there now that are finally adoring what they're doing and trying to learn from it, and it does a lot for our scientists, it does a lot for our advocates as well. But what I came up here for is three things. One, this is one of the best panel presentations I've seen in terms of what we need to know as advocates and how we build our advocacy. I call it a career, so to speak, and passionate career. And I also want to thank you. My gosh, Jennifer, you've done such a great job today of explaining very complex information in a very easy, accessible manner. Wonderful slides. So I want to congratulate you on that.
Yes.
Thank you. And you're so animated about it. It's great.
Yes.
So, the one thing I wanted to ask you, because I didn't hear you say anything about it, have you been integrating into your thought processes, how do we identify biomarkers that help tell us who's going to benefit, who's not, and also who might be harmed?
Yeah, those are great questions. Yes, the PARP study the AXLAP trial that I mentioned that we're starting that tests the CSF1R inhibitor with PARP inhibitor. We have biopsies and blood and stool and serum banked from those very generous patients, and so we will do all of the analysis. And then I mentioned we just got a huge grant to actually do the work, and so our question, and we hope to have 30 patients. So I think it should be enough numbers to start to get an idea, but that is exactly what we need to understand in the tumor biopsy at the baseline. Which types of macrophages are there when there's response, and which ones are there when there's no response to that combination, and then extend that beyond just that one trial.
One other thing, please. As you're also going through this trial, please try to capture and measure things that mean something to patients.
Yep.
There was a recent study done by Dr. Ross Kamage, who's a lung cancer researcher who was diagnosed with lung cancer. Mm-hmm. And he was appalled by what he was finding out as a patient that he didn't have any idea of when he was an oncologist researcher. And so he did a study about what we are capturing in terms of side effects and toxicities. And what he found is we aren't capturing much of anything that means anything to a patient. Yeah. What we see as tolerable is something that patients are often enduring despite losing their real life as a result of it. So I would encourage you to work with your advocates to try to find measures. And we don't think we quite have that yet, but try to find measures that help patients and future patients know what they're getting into for better or worse.
Mm-hmm. Yeah, thank you. That's very important.
So I'm Jane Perlmutter. Cheryl took my thunder. It was a great work, and you really are such a great speaker. You energized, I suspect, everyone in the room. But my question is, I had a red flag when you said bad macrophages, bad TREM2. And maybe I misunderstood. Are these naturally in the body and just show their way into the breast, or are they only there from breast cancer? And if they're in the body naturally and you're killing them, what harm are you doing?
Yeah.
And
They're
whatever.
super important. So I use the word bad to make it simple. I don't actually like to use that word because macrophages are very complex. I like to talk about the biology of them, but to get the point across that these are the most suppressive. They are the most dangerous to. So across all solid tumors, we find TREM2 positive macrophages. In every solid tumor, the presence of these correlate with worse clinical outcome. They're not found in normal tissue. So it's unique to the tumor, and it has to do with the signaling, that way that these macrophages use energy through lipid metabolism. That's what fuels them to continue to secrete things that inhibit T cells, which makes it an attractive candidate therapeutically. Yeah.
So they're misguided.
Hi, I'm Carolyn Royce from Colorado. First presentation I've seen as an Alamo Scholar this year. So wow, it was great. And I guess when I saw the trial outline, it mentioned the RESIST measurement criteria. And my question is there other ways to measure progression for those type, because RESIST will exclude a lot of non-measurable tumors like lobular, et cetera.
Yeah, that's a very clinical question for the PhD immunologist. So I don't know the answer to that, but I can find out and get back to you. Yeah.
Hi, thank you for your presentation. It was wonderful. My name is Ivis Phoebus Sampaio. I'm a project lead graduate, previous NBCC board member, retired chief officer of diversity and inclusion at Share Cancer Support, and currently the patient advocate for the Disrupt project. I just wanted to make two comments. First, you're absolutely right about what's going on with the patient advocates and how we're being asked to be a part of something, and they want to write the letter, as you mentioned. And, you know, we all have the right to say no. And I think I want to make sure that the new advocates understand that. I had an experience where a wonderful researcher reached out to me because she wanted, her project was focusing on the Latino community. And she, after they wrote it up, after they started it, then they couldn't get enough, so she reached out to me then. So I told her very nicely that I appreciate, you know, her reaching out to me, but that obviously she needed to reach out to me from the very beginning, because as she gave me the information, I told her everything that was wrong with it and why she wasn't getting anybody. We continued our relationship. Years later, she reached out to me when the concept was in her mind. We went through it, we broke it down, and she's become one of the best research advocates for patient advocates. So your words do matter. And secondly, I was gonna ask, once, I know this is right now in the lab, but as you start to grow with this and the ideas, I would like you to continue to include diversity within that, because you might find some differences. And plenty of times I've worked with researchers that say, you know, when I'm, you know, they used to know me as Ivis, is it available in Spanish, Sampaio. But the point is that if you start from the very beginning, thinking ahead, you know, and working with the patient advocates and saying, okay, one thing, what's missing? You know, do we have a diverse community? Do we have diversity in age, in location? All of these things from the very beginning, so that then you don't have to turn around and say, oh, but that's another good trial that we could start. No, do it all from the very beginning, and advocate for the funding and get the patient advocate that will be there to say, hey guys, stop spending our money the wrong way, get it done right.
That's great.
Thank you.
Yeah. Well,
I do like pushing back on letters. As a reviewer of grants, when I read a letter that's not a sincere relationship, you can tell. You can tell when a patient advocate has just met the researcher, you know, the week before, and it's just not a genuine relationship that's going to be beneficial at that point. Yeah.
and when Jennifer's work goes to clinical trial, there's a physician starting that clinical trial who has another team of patient advocates from the very start. We're forming that trial with her from the concept.
Yeah.
Good morning. My name is Jill. I'm here from Vermont as a brand new patient advocate. It's a little weird. I work for industry in the pulmonary space, and so I've worked with those patient advocates for years, and I didn't ever think that I would end up on the other side of things. But my question for you is, you talked very specifically about solid tumors. And do these bad macrophages, I know you don't like the word, but these bad macrophages, are they also highly present in patients, whether early stage or metastatic, that don't have solid tumors and have spread to, other parts of the body and don't involve any solid tumors?
Are you referring more to metastatic lesions or to like hematologic, like blood cancers?
It could be blood cancers. It can be metastatic spread to bone marrow. I mean, where there are not, not all patients have solid tumors,
Sure.
Whether it's early stage, whether it's an occult breast cancer or, whatever. But in both scenarios, metastatic versus earlier stage, but is this still a problem even when there are no, quote, solid tumors?
Yeah, so great question. So most of what I talked about today was the primary tumor, this primary solid tumor. When we evaluate, for example, from breast tumors, the liver mets or brain mets or lung mets, TREM2 macrophages are highly infiltrated. In fact, there was a paper maybe last year by another group that characterized the lung metastases as being surrounded by TREM2 macrophages. So T cells couldn't get in because it was just surrounded by, which is exciting biologically to understand that, right? And to know that we can maybe target those. In terms of ascites or ovarian types of things, yes, they're highly prevalent there. In hematological malignancies, we do see them in those sites. Very few people work on macrophages. Rosie Kaplan's an example, but and so everybody at Dana-Farber and Brigham and the Harvard system has asked my team can you analyze our macrophages? What's going on in our macrophages in their favorite cancer type, right? And we've done them across both blood cancers, solid tumors, metastatic setting, and they're there in high numbers. Yeah.
Hi. First of all, thank you, because you explained it for us, for me at least, who doesn't know the science. So I want to thank you. But my question is with this, I know it's a lot going on. With the AI, we hear, I'm not an IT, but I hear a lot, amazing thing. So integration coming soon, or I didn't hear anything, so.
Yeah, so are you like maybe pathology-based AI or?
Yeah, to combine and.
Sure. So when I showed you that colorful, I called it a UMAP, it was the multi-dimensional data that was all different colored spots on the graph. There's 16 different types of macrophages that we can find through single cell analysis. We can't do single cell analysis on everyone's tumor, but every patient has a biopsy that's on a slide, right? So yes, there is a lot of interest in using AI to understand, can we find those 16 types of macrophages in that slide, that's on the glass slide that's used for clinical diagnosis. It's difficult to train those models to find these different types of macrophages. I don't think it's impossible. One advantage we might have with these lipid macrophages that I've sort of defined these bad macrophages as lipids are fat droplets. We actually can see that morphologically on the slide. So it's possible we may be able to identify these using AI. Yeah.
We have someone on this side.
I think she's been standing.
I'm Connie Tilley. I'm from Illinois. I'm an Alamo scholar and project lead graduate. Actually, I got two questions. One is, have you done any studies with human organoids regarding this technology? And the second is, once you're infused with this, I guess I'm calling it CAR-M.
Great.
Then, say it wipes out the tumor cells, how are the tumors themselves, how do you keep it from regenerating through stem cells?
Yeah, okay, so First question about organized, great question. So we're not doing the organized in our lab, although I think we are going to start doing them for that CAR project. My colleague Tyler Miller out at in Cleveland has built this beautiful breast brain met program using brain met organoids and with matched blood, and can infuse the blood so that the downside sometimes to organoids is that the immune cells die very fast. The tumor cells stay within this organoid, so the organoids, right, are pieces or chunks of patient tumors that are cultured. The immune cells start to die out, but you can sort of repopulate them with the matched blood from the patient. So he's doing that very successfully. We're working on a collaboration to get some of that started in our lab, so it's a great setting, especially for breast cancer and especially for HR positive disease, where there are no mouse models that really recapitulate the disease. Not that mouse models are perfect, but and then in terms of the CAR monocytes, our goal is to get T cells to be activated. And our strategy is get rid of the bad macrophages so that the T cells can come into the tumor and be activated. And I had mentioned that an advantage of a T cell is that it has memory. So if you can get a T cell to get to the tumor and be activated, it should have long-term memory, have that durable response against metastatic sites, or if there's, the primary tumor comes back, or so that's the goal.
Okay, we have time for one more question.
Yeah, okay, thank you. This question is actually piggybacking off of what Jane asked. Her question kind of stimulated some things for me. So thinking about TREM2, CSF1R, you mentioned that those macrophages are not present in the body normally. So are those silent genes that somehow get turned on? And at what stage do they turn on? What causes them to turn on those genes to start producing those bad macrophages?
Yes. I'm happy to have a deeper scientific conversation, but it does have to do with the things that the cancer cells are producing and secreting, called cytokines. And those, actually, if you just put a macrophage in a dish and use those same cytokines, it will turn on these CSF1R, TREM2, CD36. So it has to do with the tumor and how it recruits monocytes, which become macrophages, and so it's all of that signaling directly from the tumor. But you're right, it's the TREM2 is in the cell. It's just it is induced for expression in that setting. Yeah, it's a really good question.
What are the specific cytokines?
GM-CSF.
Are we? Sorry. Well, thank you, everyone. Obviously, very engaged conversation. Thank you, Dr. Guerriero, for enlightening us. And keep your eye out for more training from NBCC to really connect advocates to researchers. So thank you, everybody.